Okay, today we'll start with the UniProt, that is the universal protein resource. And I'm starting with some questions and that I'm showing in a small, and I'm showing a small hand-on session on this UniProt. So what is UniProt? So UniProt is a universal protein resources. It is a comprehensive, high quality and freely accessible database of protein sequence and functional information, and it is widely used in the bioinformatics and molecular biology and proteomics. So UniProt was formed in 2002 through a collaboration of three leading bioinformatics institute, that is European Bioinformatics Institute, that is EBI, or EMBL EBI, the Swiss Institute of Bioinformatics, that is SIB, and the Protein Information Resource, that is PIR. So what are the general features of the UniProt? So UniProt is a protein sequence that contains the sequence with wide range of organisms. The functional annotations, which provides the detailed information about the protein function, domains, its active sites, and its biological roles. The third one is the protein structure and interactions, which can link down to the known 3D structures of proteins and their protein interactions, and the cross references, which is connect to the other databases like PDBs, KEGG, gene ontology, and other etc. The UniProt is basically a reviewed or unreviewed, that is the validated and non-validated data both. So the SwissProt, that is the curated and manually retrieved entries from the validated data, which is published in the work or experimentally validated work. And UniProt TREMBL, which is a computationally analyzed unreviewed entries. Next, we can see in the UniProt section or UniProt database. So what is, we can find the main components that are the UniProt KB, which is a knowledge-based protein sequence and function, which is based on SwissProt and TREMBL. UniRef, that is the cluster of sequences to reduce the redundancy of the proteins and the speed up our searches. The UniPARC, that is the archived all the protein sequence, which is to track the sequence history. And UniMES, which is focusing on the metagenomics and environmental sequences. So what is now the UniProt KB? The UniProt KB is the main component of UniProt database that provides the detailed and high quality information about the protein sequence and their biological functions. So which kind of protein you can choose, their information and their protein sequences and their biological functions is in detail in the UniProt section. The UniProt KB is divided in the based on two sections, that is the UniProt KB SwissProt, which is manually retrieved and curated entries with the reliable annotation. That means the proper literature evidence is present. And UniProt KB, that is the TREMBL, which is automatically annotated or unrevised entries. These are basically computationally curated records with no literature references, or it is being represented as the other databases. Next, we can see the proteome database. So proteome is a section of UniProt, and it is a complete set of proteins expressed by the genome of an organism. So the whole organisms and its proteome means its proteome IDs, and that is the full portions of full protein structures of this organism actually. What are the key points of this? Each organism had a specific proteome ID that I will discuss in the hands-on portion. The proteomes include the all known and predicted proteins from the whole genome set, and they are useful for the comparative genomics, functional annotation, and in the evolutionary studies. The uniprot proteome is also classified based on the two portions. One is the reference proteomes, which is well studied and high quality sets, and another is complete proteomes, which is a full set of predicted proteins from an organism. The next one is the UniRef, which is the uniprot reference clusters. The uniprot reference clusters is a part of the uniprot database, which provides the non-redundant clusters of the protein sequence, and which can speed up the sequence searches and reduce the data redundancy. It is have the 3 levels of clustering. One is and it is based on the percentage identity actually. That is UniRef 100. It is all sequence from the uniprot with no clustering. UniRef 90, cluster of the sequence is greater than equals to 90% of the sequence identity, and first one is a 100% sequence identity. The UniRef 50, which cluster the sequence with the greater than equals to 50% sequence identity. What is the purpose of this UniRef? So makes the sequence searching faster and more efficient for our data search, and reduce the redundancy of the large scale analysis. So you can say you can search for the searches for the 100% sequence identity. So you only search for UniRef 100%, right? So useful, and the next one is the useful for the comparative genomics and evolutionary studies also. Next, we can see the UniPARC. There is a sequence archive, and the sequence archive is in the uniprot that stores all the unique protein sequences regardless their origin or the annotation. Without this, there are many kinds of organisms and their protein sequences are present in this UniPARC. What are the key features of that? It tracks the every versions of the protein sequence across the time, avoids the duplication of its unique sequence, that gets a stable UniPARC ID, and does not contain any functional annotation, and only the raw data is available. So there is no annotation present. Excuse me. So useful for the, so what is the usefulness of this thing? So useful for the sequence history tracking, the version comparison, and the cross reference with the other databases. So now our what is the basic difference between the uniprot and PDB here? So uniprot, basically the database contained with the protein sequence and functional information of that particular protein. The type of data is available in amino acid sequences, protein function, domains, pathways, gene names, disease associations, localizations, and etc. And its format is basically the text-based biological information. That is the basically FASTA format. And the type, the data is curated based on the two portion, that is the SwissProt, that is the reviewed one, and the unreviewed one is the TREMBL. And it is useful to functional annotation like the bioinformatics, genomics, and sequence analysis. This kind of work we can consider from this database. And the PDB, we can see the 3D structure of the proteins and nucleic acid we can get from the PDBs, and the type of the data we can obtain from this is the atomic level, that is the 3D coordinate data, the structural informations of extracrystallographic NMR study or cryo electron microscopy. This kind of information or data we can gathered or found from the PDBs. What are the formats? Basically, it's a 3D structural files, that is the example .pdb or .cif format we can get from this, and the data is curated experimentally validated form only, not the predicted models. There is no predicted models present in the PDBs, and it is used in the basically structural biology, the drug design, and molecular dynamics, or sorry, molecular modeling, and so on. So now there is some analyzing tools which is present in the uniprot. So what are those? These are basically BLAST. There is the basic alignment, basic local alignment search tool, Align, retrieve the ID mapping tool, the peptide search, the UniProt REST API or SPA RQL. So what is the BLAST? It comprises this kind of informations or this kind of analyzing tool we can discuss in the later sections, and with the detailed section of this portion in our next classes. But as a preliminary knowledge, we can see the BLAST, which is comprises of the protein sequence of UniProt, and it identifies the homologous structures or homologous or similar protein structures actually. And the align is from the multiple sequence alignment, that is the portion which is the sequence same or the comparing conserved regions of the selected search of any kind of proteins actually. The mapping tool, it converts the IDs from other database, that is the PDBs, Ensemble, or other UniProt IDs to changes into the UniProts and integrate into the UniProt database. The next one is the peptide search, which is the peptide fragments within the UniProt sequence and useful in the mass spectrometry and proteomics workflows. And the SPA RQL, which is basically used in the advanced users and the developers to customize their data queries according to their requirements. Now, so what is the significance of this UniProt? So it provides the central resources for the protein sequence and the functional data. So that can offers the manually retrieved or high quality annotations in the SwissProt. The details, protein functions, domains, its active sites, its pathways, its interactions with other portions or genes. It supports the biomedical research, the drug discovery, and diagnostics as well. It is the cross-link with the major databases for integrated the biological insights and the compatible with the bioinformatics tools for the analysis and the predictions. So the UniProt enables the proteome comparisons and the evolutionary studies for further research works and freely accessible for the researchers in worldwide. So this is the basic information about the UniProt basically, and now we can move to the hands-on of this UniProt. So we open our tab. Now we can search UniProt. Here is the web page link of the UniProt, and go and click this one. Here, see the portions of the UniProt. As we can show in our slides or tell into our slides, there are 4 main portions in UniProt. That is the proteins, that is UniProt KB, that is UniProt knowledge base, the proteomes, the protein clusters, that is the UniDevs, the sequence archive, that is the UniPARC. So what is the UniProt knowledge base? So this is the portion of the knowledge base portion. Here we can see that the 2 entries are present. One is SwissProt and one is TREMBL. So you can see that it is a pre-reviewed portions. That is we can talk about. It is the experimentally validated or the literature evidence data. That is the portions. That is the 573230 NTs. And another one is unreviewed. That is the TREMBL, which is computationally based, or this is not actually the literature evidences. Okay. Next, the portion that is the species proteomes. This is the portion. There you can see that there are also reference proteomes and non-reference proteomes you can see, and the proteomes contains the unique proteome identifier. That is a specific persons or organism has a specific proteome identifier that we can see in the inner section of our UniProt. Okay. Next, we can go to the uniprot clusters, that is the protein cluster, that is the UniRef, that we can see that there are three portions. There is the UniRef 100. There are this much number of clusters is present, which is 100% identity. Another one is UniRef 90, that is the portion of 90% identity, and this is the number of clusters is present. So you can go to the README section and or the FTP section to download these files and uh, extract your data or whatever you can work with. Next, we go to the sequence archive of the UniPARC. That is the portion. It is the actually the reference portion which contains the different kinds of sequence proteins or the sequence portion. You can see this. This is the reference sequence UniPARC. This is the documentation, and there is a FASTA format. The whole FASTA format you can download from this section. Okay. So these are the basic portions or the sorry, excuse of the portions or basic three components of our uniplots, and there are some other portions. There's the latest news which can be regarding the uniplot, that you can see this portion, and this is the another one portion of the spotlight portion. You can see this portion, and then we can go to the next portion, that is the analyzing tools. So that I will discuss. I was discussed in the previous slides. That is the BLAST, the align, the search with map IDs, and search the smaller peptides also. So this kind of analyzing tools we can use in later section as a single module, and there are some uniprot data you can download or you can access by your own. That is the FTP downloads, the technical documentation, which how can these informations is produced. The programming access also there. You can access this portion, that is the REST and SPA RQL, which is specifically for the developers, which can develop this website more efficient also. And you can submit your data, or you can originate this and submit your data in these uniplots. So now, what kind of information we can get from the uniplot? Now I have discussed this. So you can see the left drop down portion. There is a uniplot KB. This is the search option. You can search by different methods. I will show you one by one. Okay. So in uniplot KB, here are different kinds of portions you can search with. You can select as uniplot KB, uniplot UniRef, or uniplot PARC, proteomes, taxonomy. As per your choice, you can search according to your relevant information. So uniplot KB is the large portions of knowledge base data. So I can search with this portion of uniplot KB. So I'm searching with a name, that is nitrogen. So I'm searching. Okay. Okay. So I'm searching the portion is MEPK1. So you can see that here is just left portion or the left side. There is a two status is defined. That is the reviewed one and unreviewed one. So reviewed one is the sweet spot, which I am discussing about. And the unreviewed one, that is the TR EMBL, which is present here. So you can select this to, this is the sort the data as per the reviewed one, that all of these portions have the literature evidence present. So now you have to search as per your requirement which kind of data you have required for your work or for your problems. So you can select the human, the only 249 data will available here. So I can select the 249, that is human section. So here only present, that is in the mitogen activated protein kinase, and there are only 249 hits that is that will given by the uniprot, and these are the portions, right? So you can see there are other portion, that is the filter by taxonomy, group by keywords, gene ontology, or the 3D structures. There are only 201 hits which have only 3D structure available. There are only 96 which have active sites available. So as per your portions, you can see all of this, right? So now we switch to the another one, that is, again we can search as uniprot. Now we can search as a, I can search previously in the section as a that is the gene name, that is MAPK1. Now you can search as so here you can search as a text format, that is a text name, that is the portion you can see that there is a, there is also hit the same kind of portions. We can search as a single name or a single gene name also. Now we can switch another portion, that is the advanced search. So how can you search with this advanced search? You can write the MAPK1, its portion is Mammalian. So mammalian portions you can search, or you can search by other names also. And then keywords is not necessary, or you can also input as a keyword and then find the search. See, there are only 4 portions or 4 available for the humans or the mammalians portion in the uniprot. So there are 235 results, but only 4 is in the reviewed section. So if you click the reviewed, there are 3 portions, that is 1, 2, 3, 4. They are basically in the mammal sections, that is human 1, bovine 1, mouse, and rat. So there are these portions of the this one, that is P2848. This is the accession number actually. You can search by also the accession number which is present in any literature. I can show you also. And then you can see that there is a present in the human, there is the entry name in the human, and this is the Bostoros, there is a bovine or a cow, and this is the mouse molluscus, there is the mouse, and there is the Rattus norvegicus, there is the rat. You can see this one. Now another one I can show you. This is the advanced search. So now you can search with P 8 See, you can see there is a portion which is present in our uniprot. There is a single one, that is a unique identity or unique accession number IDs of the MAPK1 of the humans. So that is present here, right? Okay. So now what kind of information we can see in this portion? So we can go from this, that will be helpful. There is the entry portion. There are many entries. There is the 175 entries here for the humans. And there are the specific 6-digit entry or the accession number is specifically for specific uh, proteins or the peptides or in the uniprots, right? So here is the entry name. There is the MK01 for the human. There is the protein name which we are deal with. There is a mitogen activated protein kinase 1. His other gene names or the other entries, other names for this specific protein. There is a MAPK1, PRK2. These are all of the isoforms actually. So PRK M1, PRK M2, and this. And this is the protein length of this sequence, that is 360 amino acid. So now we go to this and we see this kind of portions. We can see this. So what are the portions, or what are the another informations we can given, or we can see from this? There is the entry. What are the functional portions of this MAPK1? That is the variant viewer. What kind of variant viewer we can see? So see here the filter consequences. So what are the, this is the 360 amino acid sequence of this protein, and which one is pathogenic, which one is the predicted consequence, which one is a beginning, and which one is uncertain significance, that is shown in this, in that section, right? So here you can see that the dot ball sections, there is a green portion that is the uncertain significance of this region actually. There is the red region, that is the pathogenic or the non-pathogenic portions of this of that portion of the protein sequence, right? Next is the and here is the portions for the sequence here. Next is the portion, there is a feature viewer. So what is the feature viewer? There is a molecular processing, that is the chain. What is the length of the chain? That is, you can see that is 1 to 360 is the chain. So this is the chain portion of this portion. The initiator methionine portion is the initial portion, that is the number 1 region. There is a sequence information. Sequence information, that is what are the domains, and what are the domains is the, in the purple and these sections. These were the portions. So these are the whole domain section of this specific protein, that is a MAPK1, this form to this form. And these are the portions, these are the motif region. So motif, what is the domain and motif? Domain is the large portion of the protein section, and motif is a smaller part or the smaller regions of this protein. So these are the specific regions on the motif section, right? So here of the at another site, that is the another portion, that is the sites, that is DNA binding or active site, that is represent here. So what are those? The DNA binding sites. This region is actually shows the DNA binding regions. Okay, and there is the portion we can see that is you can see in the left section also. There is a modified residue. So what kind of residues is modified or some kind of changes is shown in that protein structures. So there are other portions of the proteomics and AlphaFold or the AlphaFold missense portions that is shown in this portion. And there are some specific features. So the protein have a secondary structure that we all know. So which kind of structural portions is available in this whole protein amino acid chain? So helix is the pink part that is shown in the diagram, and that the beta strand, that is the yellow portion of this protein region, that is shown in the yellow section, and the turn sections, that is blue sections, is shown in that portion, right? So here is the numbers, there is the 100 to 200 and the 360 portions, right? So now these are the portions of the feature viewer. So now we can switch or move to the genomic section. So there are two portions or the genomic coordinates. So what kind of things we can see? This is the chromosome numbers that is present in the, which chromosome number is present in that portion. There is a number, chromosome number 22. The assembly name, that is the GRC38. What are the number of exon? What is the exon? These are the protein coding portions of the specific peptide or the protein. So the number of exon is basically 8. And there is another isoform, that is P28482 and P28481. These are two isoforms. So here in isoform 1, that is 8 exons are present, and in isoform 2, that is 7 exons are present, right? So these are the portions where the exons are present. So 1 to 40 region A, 1 to 40 region shows in one exon number 1, that is 40 to 101 is number 2, and this, right? So these are the genomic portions. So here another one is publication and external links, that is also present. So what kind of publications is regarding with this structures, we can see that. Okay. So now, so in the entry, so now we can switch to the portion, that is the naming taxonomy. These are the functions. So which kind of function is basically present in here? Their names and taxonomy. That is the map kinase 1. That is the alternative name is ERT1, ERK2, MAPK, MAPK2, all of this. And these are the subcellular localization where the this is present in the portion. There is the information, the cytoplasm, that is the nucleus, that is the our membrane caveola. These are the cell junctions. So these are the portions we can see this peptide or protein actually, right? So there are some another portion which can see. Here is the interaction site, that is how this protein can interact with other proteins, right? So there are the list of this portion, that is the that is present in this portion. There's DUSP1, DUSP4, DUSP5, and MAPK, MAP2K1, LZTS1. So these are the interaction of other peptides or other genes of this with this peptide or protein, right? So here the next section is the structure. So what are the structures basically? So structure is a portion that is I can discuss in my slide. That is the you can we can find the 3D as well as the text-based model, both present in this UniProt. So we can see the different kinds of PDB IDs or PDBs which can found this MMPK1, right? So there are the PDB IDs and their resolution, their method, that is extracrystallographic or NMR, whatever it is, that is present here. So now we can show the family, right? So as we can see that the previous section, what are the domains and motifs are present in that portion. So here the 25 to 313, this portion is the mainly domain region of the protein kinase of this MAP kinase 1, right? You can see that this is the domain portion. And the motifs, there is the 185 to 187, this is the functional portion. The 318 to 322, there is another portion. 327 to 333, this is another portion, right? And next is the sequence and its isoform. So what are the portion? That is a text-based portion which we can find out from this, that there is a FASTA format or the whole protein sequence from 1 to 360. We can find from there, and we can download this portion and copy it from this and paste it and save it for the future process or the further next process, right? Now we can form this, and there is some isoforms that we can see in the previous portion. That is the P24821 and 24822. These are the uniform portions, and there are similar proteins which is found in that portion, that is present in also in human, also in mouse and other portions. So you can search by your own, and if any query about this, you can tell me in our next session. And you can also have a 430 question answer session for your assessment or the videos which is uploaded in the Vula, and you can ask the and upload it in the Vula platform, and you can ask the question answer sessions in the 4:30 p.m. and you can ask your query about there, and your assessment submission date is tomorrow. So everyone who will complete your assessment, please upload in the Bula platform. So this is all from the UniProt. So the analyzing tools we'll discuss later, a later section of our next classes. Okay. So thank you all.